Audiobook NCRT Class 11th Chapter Number 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that was scientific, but born out of a need to use organism for our own use, for food, for shelter and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs and herbs. He also divided animals into two groups, those which has red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdoms were developed that included all plants and animals respectively. This system did not distinguish between the eukaryote and the prokaryote, between the unicellular and the multicellular organism, and photosynthetic, that is green algae, and non-photosynthetic, that is fungi organisms. Classification of organism into plant and animal was easily done and was easy to understand, but a large number of organisms did not fall into this category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationship, etc. Classification system for the living organism have hence undergone several changes over the time. Though plant and animal kingdom have been a constant under all different systems, the understanding of what groups or organisms be included under these kingdoms have been changing. The number and the nature of other kingdoms have also been understood differently by different scientists over the time. Table 2.1 Characteristic of the five kingdom according to R.H. Whitaker. Characters are number 1 cell type, and there are five kingdom according to evolutionary relationship. First is Monera, second Protista, third Fungi, fourth Plantae and fifth Animalia. Cell type: Monera is prokaryotic, Protista is eukaryotic, Fungi is also eukaryotic, Plantae is eukaryotic and Animalia is eukaryotic. Number 2 character, cell wall. In Monera, cell wall is made up of non-cellulosic cell wall, that is made up of polysaccharide and amino acids. In Protista, it is present in some organisms. In fungi, cell wall is made up of chitin. In Plantae, cell wall is made up of cellulose, and in Animalia, cell wall is absent. Number 3 character, nuclear membrane. In Monera, nuclear membrane is completely absent. In Protista, fungi, Plantae and Animalia, nuclear membrane is present and that's why they are eukaryotic. Number 4 character, body organization. In Monera, body is made up of only cells, that's why cellular organization. In Protista, cellular organization. In fungi, it is multicellular and made up of loose tissue. In Plantae, tissue and organ level is present. In Animalia, tissue, organ or organ system, all three levels are present here. Number 5 character, mode of nutrition. In Monera, most of the prokaryotes are autotrophic. They can be chemosynthetic and photosynthetic, and they are heterotrophic also, and there they can be saprophytic and parasitic. Protista mostly autotrophic and they are mainly photosynthetic and heterotrophic. In fungi, most of the organisms are heterotrophic, they are saprophytic or parasitic. Plantae are mostly autotrophic and shows photosynthesis process. In Animalia, most of the organisms are heterotrophic, they shows holozoic mode of nutrition and they are saprophytic in nature. So, R.H. Whitaker in 1969 proposed a five-kingdom classification. The kingdom defined by him were named as Monera, Protista, Fungi, Plantae and Animalia. The main criteria for classification used by him included what? That included cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationship. Table 2.1 gives a comparative account of different characteristics of the five kingdoms as I told you before. The three domain system has also been proposed that divides the kingdom Monera into two domains. Domain is greater than kingdom, leaving the remaining eukaryotic organism in the third domain and thereby a sixth kingdom classification. You will learn about the system in detail at higher classes. So Monera is divided into further two different domain, and that's why there are three domains and six kingdom. Let us look at this five kingdom classification to understand the issues and consideration that influenced the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm and angiosperms under plant. The characters that unified this whole kingdom was that all the organism included had a cell wall in their cells. This placed together group which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, which is the cyanobacteria, with the other group which were eukaryotic. It is also grouped together the unicellular organisms and the multicellular ones. Says, for example, Chlamydomonas and Spirogyra were placed together under the algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plant. Though they also showed a characteristic difference in their cell wall composition, the fungi had a chitin in their cell walls while the green plant had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, that is kingdom fungi. All prokaryotic organisms were grouped together under the kingdom Monera, and the unicellular eukaryotic organisms were placed in the kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, which was earlier placed in the algae within the plants and both having the cell wall, with Paramecium and Amoeba. Amoeba was earlier placed in the animal kingdom which lacks cell wall. It has put together organisms which in earlier classification were placed in different kingdom. This happened because the criteria for classification change. This kind of changes will take place in future to depending on the improvement in our understanding of characteristic and evolutionary relationship. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and the reproductive similarities, but it is also phylogenetic, that is based on evolutionary relationship. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, will be dealt separately in the chapter number 3 and chapter number 4. 2.1 Kingdom Monera. Bacteria are the sole member of the kingdom Monera. They are the most abundant microorganism. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in the extreme habitats such as hot springs, deserts, snow and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as a parasite. Bacteria are grouped under 4 categories based on their shape. The spherical shape, coccus, plural is cocci. The rod shape, bacillus, plural is bacilli. The coma-shaped Vibrium, plural is Vibrio, and spiral shape Spiralium, and the plural is Spirilla. All are mentioned in the diagram 2.1 or figure 2.1, that is bacteria of different shape. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group shows the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substances. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotroph, that is, they depend on other organism or on dead organic matter for food. 2.1.1 Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophiles, hot springs called as thermoacidophiles, and marshy areas, for example methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of the several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall, and if motile, they have a flagella called flagellum. The cyanobacteria, also referred to as blue-green algae, have a chlorophyll A similar to green plants and are photosynthetic autotroph. The cyanobacteria are unicellular, colonial or filamentous, freshwater and marine or terrestrial algae. The colonies are generally surrounded by a gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in the specialized cells called heterocyst. For example, nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as the nitrate, nitrite, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are the most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotic, fixing nitrogen in legume roots, and some are pathogens causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacteria to the other. The mycoplasma are the organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. In figure 2.2, nostoc example is given, which is a filamentous blue-green algae. There are two types of cell. One is vegetative cell, which shown in the green color, and there is another special type of cell, that is heterocyst, which is given in bluish or purplish color, and whole structure is covered by a gelatinous sheath. It is a filamentous algae. In figure 2.3, there is a structure in which bacteria divide by the binary fission. First of all, cell wall divides and starts to form furrow, and one single cell of bacteria divides into two different bacterial cells. It is a type of binary fission. 2.2 Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protists to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellate, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other, dealing with plants, animals, and fungi. Being eukaryotes, the protistian cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protista reproduce asexually and sexually by a process involving cell fusion and zygote formation. Chrysophytes 2.2.1. This group includes diatoms and golden algae, that is desmids. They are found in the fresh water as well as in the marine environment. They are microscopic and float passively in water current, that is they are considered as plankton. Most of them are photosynthetic. In diatoms, the cell walls form two thin overlapping shells which fit together as in a shop box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind a large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as a diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in the oceans. 2.2.2 Dinoflagellate. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigment present in their cells. The cell wall has stiff cellulosic plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often, red dinoflagellates, for example goniolates, undergo such rapid multiplication that they make the sea appear red, which is known as red tides. Toxin released by such large number may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in the stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle which makes their body flexible. They have two flagella, one is a short and the long one. Though they are photosynthetic in presence of sunlight, when deprived of the sunlight, they behave like heterotrophs by predating on the other smaller organisms. Interestingly, the pigments of the euglenoids are identical to those present in the higher plant. For example, euglena in figure 2.4b. 2.2.4 Slime mold. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies, bearing spores at their tips. The spores possess true walls. They are extremely resistant and survive for many years, even under the adverse conditions. The spores are dispersed by air current. 2.2.5 Protozoans. All protozoans are heterotrophs and live as a predators or parasite. They are believed to be primitive relative of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, seawater and moist soil. They move and capture their prey by putting out pseudopodia, which are the false food, as in amoeba. Marine forms have silica cells on their surface. Some of them, such as ant amoeba, are parasite. Flagellated protozoans. The members of this group are either free-living or parasitic. They have a flagella. The parasitic forms cause disease such as the sleeping sickness, for example trypanosoma. Ciliated protozoans, these are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of the rows of cilia causes the water laden with food to be steered into the gullet. For example, paramecium in figure 2.4d. Sporozoans, these includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite which causes malaria, a disease which has a staggering impact or effect on human population. In figure 2.4, A figure is for dinoflagellate, B figure is for euglena, C figure is for slime mold and D figure is for paramecium. Kingdom fungi 2.3. The fungi constitute a unique kingdom of the heterotrophic organisms. They show a great diversity in the morphology and the habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstool are also fungi. White spots seen on mustard leaves are due to parasitic fungi. Some unicellular fungi, for example yeast, are used to make beer and bread. Other fungi causes disease in plants and animals. For example, wheat rust causing Puccinia is an important example. Some are the source of antibiotics, for example penicillium. Fungi are cosmopolitan and occur in air, water, soil and on humans and animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent the food from going bad due to bacterial or fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their body consists of long, slender, thread-like structures called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called cenocytic hyphae. Others have septae or cross walls in their hyphae. Their cell walls of fungi composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from the dead substances and hence are called saprophyte. Those that depend on the living plant or animals are called parasite. They can also live as symbiotic in association with algae as lichens and with the roots of the higher plant as mycorrhizae. Reproduction in fungi can be take place by the vegetative means, fragmentation, fission and budding. Asexual reproduction is by spores called conidia or sporangiospores or zoospores, and sexual reproduction is by oospore, ascospore and basidiospores. The various spores are produced in distinct structures called fruiting bodies. The sexual cycle involves the following three steps. Number 1. Fusion of protoplasm between the two motile and non-motile gametes is called plasmogamy. Number 2. Fusion of two nuclei called karyogamy. Number 3. Meiosis in zygote result in haploid spores. When a fungus reproduces sexually, the two haploid hyphae of compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately result in the diploid cell. However, in other fungi, for example Ascomycetes and Basidiomycetes, an intervening dikaryotic stage, that is N plus N, that is 2 nuclei per cell, occurs. Such a condition is called dikaryon and the phase is called dikaryophase of fungus. Later the parental nuclei fuse and the cells become diploid. The fungi forms fruiting bodies in which reduction division occurs, leading to formation of haploid spores. The morphology of the mycelium, mode of spore formation and fruiting bodies form the basis for the division of the kingdom into various classes. 2.3.1 Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasite on plants. The mycelium is aseptate and cynocytic. Asexual reproduction takes place by zoospore, motile, and by aplanospore, non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by the fusion of two gametes. These gametes are similar in morphology, isogamous or dissimilar, anisogamous or oogamous. Some common examples are mucor, figure 2.5a, rhizopus, the bread mold mentioned earlier, and albugo, the parasitic fungi on mustard. 2.3.2 Ascomycetes. Commonly known as sac fungi, the Ascomycetes are mostly multicellular, for example penicillium, or rarely unicellular, for example yeast, that is Saccharomyces. They are saprophytic, decomposer, parasitic or coprophilous, that is growing on dung. Mycelium is branched and aseptate. The asexual spores are conidia produced exogenously on the spatial mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci, singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are Aspergillus, claviceps and Neurospora. Neurospora is extensively used in the biochemical and the genetic work. Many members like morels and truffles are edible and considered delicacies. 2.3.3 Basidiomycetes. Commonly known form of Basidiomycetes are mushrooms, bracket fungi or puffball. They grow in soil, on logs or on tree stems and in living plant body as parasite, for example rust and smut. The mycelium is branched and septate. The asexual spore are generally not found, but a vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotype. The resultant structure is a dikaryotic, which ultimately give rise to basidium. Karyogamy and meiosis take place in basidium, producing 4 basidiospores. The basidiospores are exogenously produced on the basidium. Plural is basidia. The basidia are arranged in fruiting body called basidiocarps. Some common members are agaricus, that is mushroom, and Eustylago smut and Puccinia rust fungus. 2.3.4 Deuteriomycetes. Commonly known as imperfect fungi because only the asexual or vegetative phase of these fungi are known. When the sexual form of these fungi was discovered, they were moved into the classes they rightly belonged to. It is also possible that asexual and vegetative stage have been given one name and placed under deuteriomycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of the deuteriomycetes. Once perfect sexual stages of the members of deuteriomycetes were discovered, they were often moved to ascomycetes and basidiomycetes. The deuteriomycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Colitotrichum, and Trichoderma. There are some in figure 2.5. Fungi. First is A is mucor, second one is for aspergillus, and third one is for agaricus. 2.4. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as the insectivorous plant or parasite. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cuscuta is a parasite. The plant cell have an eukaryotic structure with prominent chloroplast and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae includes algae, bryophyta, pteridophyta, gymnosperm, and angiosperm. Life cycle of plant has two distinct phases, the diploid sporophytic and the haploid gametophytic. That alternate with each other. The length of the haploid and diploid phases and whether these phases are free living or dependent on others vary among different groups in plants. This phenomena is called alternation of generation. You will further details of this kingdom in chapter number 3. 2.5. Kingdom Animalia. The kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack their cell wall. They directly or indirectly depend on the plants for food. They digest their food in an internal cavity and store food reserves or as glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Some of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Salient features of various phyla are described in chapter number 4. 2.6. Viruses, viroids, prions, and lichens. In the 5 kingdom classification of Whitaker, there is no mention of lichens in some acellular organisms like viruses, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effect viruses can have on us, even if we do not associate it with our conditions. Viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structures outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? Virus means venom or poisonous fluid. Dmitry Ivanovsky, 1892, recognized certain microbes as casual organisms of the mosaic disease of tobacco. Figure 2.6a. These were found to be smaller than bacteria because they passed through bacteria-proof filter. M.W. Bezernik in 1898 demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and named the new pathogen virus and called the fluid as Contagium vivum fluidum, infectious living fluid. W. M. Stanley in 1935 showed that viruses could be crystallized and crystals consist largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasite. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No virus contains both RNA or DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single-stranded or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are usually double-stranded DNA viruses. The protein called capsid, made up of small subunits called capsomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause disease like mumps, smallpox, herpes, and influenza. AIDS is also caused in humans by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing, and stunted growth. Viroids. In 1971, Theo Dinier discovered a new infectious agent that was smaller than the viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. This agent was similar in size to viruses. These agents were called as prions. The most notable disease caused by the prions are Bovenger's-Pomm encephalopathy, commonly called mad cow disease in cattle, and its analog variant is Creutzfeldt-Jakob disease CJD in human. Lichens are symbiotic association, that is mutually useful association between the algae and the fungi. The algal component is known as phycobiont and the fungal component is known as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorbs mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicators, that is, they do not grow in polluted areas. In figure 2.6, number 1 figure is A, tobacco mosaic virus, in which genetic material is a single-stranded RNA and the body is made up of small protein units capsomere which form capsid as polymer. In number 2, B structure bacteriophage, there are different body parts. First part is head, second is collar, third is sheath, and the fourth is tail fibers. With the help of these tail fibers, this bacteriophage can attach with the bacterial cell surface. In head, there is mostly double-stranded circular DNA. So that's why it is a bacteriophage which can feed upon a bacteria. Summary. The biological classification of plants and animals were first proposed by Aristotle on the basis of simple morphological characters. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationship. In the five-kingdom classifications, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-celled eukaryotes such as chrysophyte, dinoflagellates, euglenoids, slime molds, and protozoans. Protists have defined nucleus and other membrane-bound organelles. They reproduce both sexually and asexually. Members of kingdom Fungi show a great diversity in the structures and the habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual mode of reproduction. Phycomycetes, Ascomycetes, Basidiomycetes, and the Deuteriomycetes are the four classes under the kingdom Fungi. Basidiomycetes and deuteriomycetes are the four classes under this kingdom. The Plantae include all eukaryotic chlorophyll containing organisms. Algae, bryophytes, pteridophytes, gymnosperm and angiosperms are included in this group. The life cycle of plant exhibit alternation of generation, gametophytic and saprophytic generation. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of these organisms is holozoic. They reproduce mostly by asexual mode. So, some acellular organisms like viruses and viroids are, as well as the lichens, are not included in the five kingdom system of classification. Thank you.